So we know that the nephron has got two parts, the glomerulus, the renal tubule. The glomerulus is nothing but the afferent arteriole, and the renal tubule consists of the Bowman's capsule, the PCT, the Henle's loop, as well as the distal convoluted tubule. So we have already discussed about the glomerulus. So let's begin discussing the renal tubule. So the renal tubule begins with a double wall cup-like structure called Bowman's capsule. The Bowman capsule encloses the glomerulus. Now, if we consider the Bowman's capsule and the glomerulus together, then the structure that is formed, we refer it to as the Malpighian body. So what is Malpighian body? Malpighian body is a combination of the glomerulus as well as the Bowman's capsule. So you know that the glomerulus is found within the Bowman's capsule. So these two structures together constitutes the Malpighian body, or also known as renal corpuscle. So glomerulus along with Bowman's capsule is called the Malpighian body or renal corpuscle. And here you can see the renal corpuscle is actually the site of fluid filtration, or where exactly the filtration of blood occurs. So now let's move on to the next part of your nephron, that is the proximal convoluted tubule, or in short we can call it as PCT. So the tubule continues further to form a highly coiled network called as the proximal convoluted tubule. Now why is it called as proximal convoluted tubule? Because if we take the Bowman's capsule as the reference, or this initial part of the nephron, we find that this coiled structure, which is shown here in red, is your PCT. And what is PCT stands for? Here P stands for proximity. Proximity means close or nearby, right? So which is that tubule which is close to the Bowman's capsule? It is your proximal convoluted tubule. And similarly, if we have a look at the tubule that we have here, which is in green color, which is far away from the Bowman's capsule, it is your distal convoluted tubule. Because this tubule, that is your DCT, is far away from the Bowman's capsule, hence we refer it as distal convoluted tubule or DCT. So once we have observed the PCT, this PCT slowly starts to descend and becomes thin. And this thin tubule that descends down and then ascends down tend to form a U-shaped loop-like structure, and this structure is what we refer as the loop of Henle. So the next part of the nephron is the Henle's loop. So it appears like a hairpin, right? So a hairpin shaped Henle's loop is the next part of the tubule, which has a descending as well as an ascending limb, which has a descending as well as ascending limb. So if we have a look once again in the diagram, you can observe that the loop of Henle first descends. So this is the descending limb, and then it takes a U-turn, and then it goes up, then it ascends. So this is the ascending limb. So the loop of Henle can be categorized into the descending limb as well as the ascending limb. Now coming to distal convoluted tubule, the ascending limb of the Henle's loop continues as another highly coiled tubular region. Okay, so the part of the nephron or part of the renal tubule which is highly coiled is actually your distal convoluted tubule. So the ascending limb continues as another highly coiled tubular region called distal convoluted tubule, or in short we'll call it as DCT. So we have discussed the structure of nephron by discussing the glomerulus, which is nothing but the afferent arteriole. Then the second part of the nephron, that is the renal tubule, which consists of the Bowman's capsule. The Bowman's capsule becomes the descends down to form the proximal convoluted tubule. The proximal tubule then gave rise to Henle's loop, and the Henle's loop then gave rise to the distal convoluted tubule. as I said earlier, that most of the nephron, they open into a common tubule or common duct, and that is the collecting duct. Okay, so let's know more about the collecting duct. So the DCTs of many nephrons open into a straight tube called the collecting duct. So remember, your nephron, if we consider the PCT, the Henle's loop and the DCT, they are highly coiled structure, whereas the collecting duct is a straight tube. Okay, the collecting duct is a straight tube. The DCTs of many nephrons open into a straight tube called the collecting duct, many of which converge and open into the renal pelvis through medullary pyramids in the calyces. So what is happening here? You can see that the collecting duct will open into the renal pelvis. You know, the renal pelvis is a funnel shaped structure. To the renal pelvis, which is a structure, which is a terminal part of the renal pelvis, it is a renal calyces. To the renal calyces, which part of the kidney is associated? It is your medullary pyramid. So your collecting duct will open into the renal pelvis through medullary pyramids in the calyces. Okay, and that is how finally the urine that is formed will be collected in the renal pelvis. Then the renal pelvis will further continue as the ureter, and finally that urine will be stored in the urinary bladder. So the Malpighian corpuscle, PCT and DCT of the nephron are situated in the cortical region of the kidney, whereas the loop of Henle dips into the medulla region. So if we have a look here in this diagram, you can see the nephrons, you can see many nephrons, and you can see that if we consider the glomerulus, the Bowman's capsule, the PCT as well as the DCT, they usually lie in the cortical region, right? This is a cortex region, right? And then this region is the medulla region, and you'll observe that the part of nephron that is the Henle's loop is the one which is actually penetrating or present in the medulla region, whereas the remaining part, that is the Bowman's capsule, the glomerulus, the PCT and the DCT, they lie in the cortex region. So the Malpighian corpuscle, PCT and DCT of the nephron are situated in the cortical region of the kidney, whereas the loop of Henle dips into the medulla. Now, based on what we have just observed in this diagram, that is, if the nephron lies, or the most part of the nephron lies in the cortex region, then such nephron will call it as cortical nephron. Similarly, you can find another nephron which is shown here. You can see that the Henle's loop is, present deep beneath the medulla region, right? Or we can say the most part of the nephron is lying in the medulla region. Then such a nephron will call it as juxtamedullary nephron. Such a nephron will refer it as juxtamedullary nephron. So based on the location of nephron in the kidney, if it is lying in the cortex region, if most part of the nephron is lying in the cortex region, then we'll call it as cortical nephron. And if we find that the Henle's loop is going deep beneath the medulla region, then we'll call it as juxtamedullary nephron. So what is cortical nephron? In majority of the nephrons, the loop of Henle is too short. Okay, in majority of the nephron, the loop of Henle is too short and extends only very little into the medulla. Such nephrons are called cortical nephrons. Similarly, as we have just observed in the diagram, in some of the nephrons, the loop of Henle is very long and runs deep into the medulla. So these nephrons are called juxtamedullary nephron. These nephrons are called juxtamedullary nephrons. So thus, we have tried to differentiate the nephron based on its location either in the cortex region or in the medulla region. If they are found in the cortex region, we call it as cortical nephrons. If they are found in the medulla region, okay, then we call it as the juxtamedullary nephron. So that brings us to the next part that we need to understand, and that is the peritubular capillary. So we have learned that the blood vessel that is entering into the kidney, that is the afferent arteriole, right? So similarly, you have a blood vessel which is leaving the kidney, and that is the efferent arteriole. So the efferent arteriole emerging from the glomerulus forms a fine capillary network around the renal tubule called the peritubular capillaries. So we come across the next part that we need to understand, and that is the peritubular capillary. So what is peritubular capillary? Basically, it is nothing but the efferent arteriole which is leaving the nephron, or which is leaving the Bowman's capsule, and it is running just beside the renal tubule. Okay, and this structure that we are observing here, we are referring it as peritubular capillaries. The function of this peritubular capillaries we'll understand when we'll be discussing the urine formation. So moving on to the next part, this peritubular capillaries, which is running just beside the renal tubule, continues further down, and it will be running just beside the Henle's loop. So the same peritubular capillaries which has descended down and is running in parallel to the Henle's loop, now we refer to this peritubular capillary as vasa recta. Okay, we refer to this peritubular capillary as vasa recta. So this vasa recta and the Henle's loop plays a very important role in a mechanism called as countercurrent mechanism, in a mechanism known as countercurrent mechanism. So what is vasa recta? A minute vessel of this network runs parallel to the Henle's loop. Which network? That is your peritubular capillaries runs parallel to the Henle's loop, forming a U-shaped vasa recta. Vasa recta is absent or highly reduced in cortical nephron. So this point is important. Vasa recta is absent or highly reduced in cortical nephron. So with this, we have concluded the structure, the location of nephron. So now we move on to the next part of this chapter, and that is urine formation. So now let's understand, by keeping the knowledge that we have gained about the structure of kidney and the structure of nephron, let's understand how the nephron will help us in the formation of urine. So to understand urine formation, we need to understand the three main steps as well as the processes. So urine formation involves three main processes. The first one is glomerular filtration, the second one is reabsorption, and the third one is secretion.